living things are made of cells, but how do those cells grow and develop? Eukaryotes have more than one cell. Their cells go through the cell cycle, where their cells grow and develop. Prokaryotes have a single cell. Their cells go through a much simpler cell cycle. We're going to focus on the eukaryotic cells. The cell cycle is a series of events the cell goes through as it grows and divides. There are 4 main phases: G1, S, G2, and M. During the M phase, the cell actually divides. The G1 phase is when the cell does most of its growing. As it grows, it creates new organelles and proteins. During the S phase, the DNA is replicated. The DNA is stored in structures called chromosomes located in the nucleus. During this phase, each chromosome is replicated so that each chromosome consists of 2 identical sister chromatids. Chromatin is coiled chromosomes. G2 is the shortest phase. The cell produces more organelles and molecules required for cell division. G1, S, and G2 phases make up the interphase, a period of growth between the cell divisions. During interphase, the cell grows and replicates its DNA in centrioles. Next is the M phase. The M phase is when the cell actually divides. There are 2 stages of the M phase: mitosis and cytokinesis. Mitosis has 4 phases. Mitosis is the division of the nucleus. The prophase is the longest phase of mitosis, up to 50 to 60% of mitosis. The chromatin condenses into chromosomes. The centrioles near the nuclear envelope separate and take up places on opposite sides of the cell, and a spindle begins to form. Metaphase often lasts only a few minutes. The chromosomes line up across the center of the cell. Each chromosome is connected to a spindle fiber at its centromere. Anaphase is the 3rd phase of mitosis. During anaphase, the centromeres joining the sister chromatids split. The sister chromatids separate into individual chromosomes and are moved apart in 2 groups. Telophase is the 4th and final phase of mitosis. Chromosomes disperse into a tangle of dense material. A nuclear envelope reforms around each cluster of chromosomes. The spindle breaks apart and a nucleus becomes visible in each darting nucleus. Mitosis is complete, but the cell is not finished dividing. Cytokinesis is the 2nd stage of the M phase. Cytokinesis is the division of the cytoplasm. The cytoplasm pinches in half. Each daughter cell has an identical set of duplicated chromosomes. In plants, a cell plate forms, and then a cell wall forms around the cell plate. But why do cells divide? Why don't they keep growing forever? Cells divide because of 2 limitations on cell growth: DNA overload and problems during material exchange. As the cell grows, more demands are placed on the DNA. The cell will have more trouble moving materials across the cell membrane. DNA overload is when the DNA of a growing cell can't meet all of the cell's responsibilities. Think about this. Suppose a small town has a library with a few thousand books. When more people move into the town gets larger. There will be more people borrowing books, and sometimes people will have to wait longer to borrow popular titles. Similarly, a large cell will have greater demands on its DNA. Material exchange problems. Materials that the cell needs, such as oxygen, food, and water, enter the cell through the membrane, and the waste leaves through the membrane as well. A cell that continues to grow larger will have difficulty getting necessary materials into the cell and waste products out of the cell because the materials will have to cover a greater distance. As the length of the cell increases, the volume increases faster than the surface area, causing the surface area to volume ratio to decrease. Think about it this way. Imagine a town with lane main street. As the town grows, more people use this street. The main street leading through the town will not increase in size. People will have more traffic as they enter and leave the town. Cell that grows will have a similar problem. The cell cycle is a series of events the cell goes through as it grows and divides. Prokaryotes go through a much simpler cell cycle, and eukaryotes go through a very complex cell cycle. During the interphase, G1, S, and G2, the cell just prepares for cell division by growing and developing more organelles. During the end phase, the cell actually divides.